Thank you so much. It's an honor to be here, and I'm sorry I missed the meeting before, but I'm very happy to be here, even though it's 80 degrees back home in San Diego. No, I'm just kidding. So I really could give this talk in one sentence and just say that the genetics research that's being done in epilepsy will change everything. And I'm now going to spend 35 minutes telling you that it's going to change everything in the way that we diagnose and treat people with epilepsy. And for those of us that are touched by epilepsy, it's not going to be fast enough. Researchers have already made wonderful, wonderful progress. But there's much work to do, and the beautiful story is that we can all work together to make it happen. So I had my introduction into the world of epilepsy in the same way that we all do. It was completely uninvited, unannounced, and threw my world completely upside down. This is my daughter, Savannah. She was born healthy and happy and typically developing until she was 2 years old. She had her first seizure. When she had her first seizure, I didn't know it was a seizure. I thought she was choking. And the paramedics came to the hospital or to the house and said, her airway is clear. There's nothing in there to suggest she was choking, but what you've just described sounds an awful lot like a seizure. This was about 20 years ago. This video was taken, and I've been in the epilepsy space for 20 years now. And I'm sad to say that I've seen more than 40,000 of these seizures in my daughter. And across a 16-year period of time, she would have many different kinds. This is just one kind here. The video is showing a number of other ones. But she also would try and fail 26 different therapies, medications, devices, diets, surgeries, and even alternative therapies. And nothing worked for her. And I think like every family who is thrown into the epilepsy world, you feel completely and utterly alone. You feel like educating yourself, meeting other people, learning about how to treat your child, whether or not you're even doing the diagnosis correctly and having the right tests done and seeing the right doctors. It's all information that is collected through clawing and searching and pushing. None of it is easy. There's nothing easy about epilepsy. And all the while that you're home trying to take care of a child who is seizing like this. I actually have hours of seizure video where she's having a very subtle seizure like this one here and thinking that she wasn't seizing. And as I was waiting for one of the big ones, the generalized convulsions, then later or years later would go back and look at the video and see that for an hour of video she actually was having a subtle seizure that I just never knew about when I was her mother. So I did the only thing that most of us can do, which is I logged them, I charted them, I kept a diary. And I think that this graph of her seizures from 1995 to 2011 is pretty representative of what epilepsy is like. It's a roller coaster. You never know what you're going to get from day to day, from minute to minute. It took them 2.5 years to diagnose Savannah with epilepsy, and the reason being is they didn't want to label her with something that would follow her for the rest of her life. It seems kind of silly when your kid's having 100 seizures a day that you would not want to call it what it is. But there's still quite a bit of discrimination and stigma attached with epilepsy, and the more that we talk about it and destigmatize it, the better off we'll be. But not only did she have anywhere between 10 to 100 seizures a day or more. I think at her best she had 5 to 10 seizures a day, and at her worst she would have hundreds and hundreds, to the point that I would stop counting and just write greater than 200. She would also go into non-convulsive status, so she'd have these status epilepticus seizures that don't resolve on their own. Basically means a non-stop seizure. And we were some of the lucky ones. Even though she would go into status weekly, she did respond to at-home medications, and this is a graph of her Diastat use, or rectal Valium used in the US, a benzodiazepine. And you can see that over time, between 2006 and 2011, the number of times she would get this medication to stop her nonstop seizures was ever increasing. And sometimes she would get it once a month, and sometimes she would get it once a week. Sometimes she'd get it 3 times a week. And it was escalating. But the epilepsy was also taking its toll on her in all of the seizures and all of the Diastat use and all of the therapies. And you could see it in every year's school picture. As you see her come home and you can see the change in her face, the loss of sparkle in her eyes, the inability to get words out, the fact that she would sleep 15, 16, 17 hours a day. And it was that point where I would go and I would ask the doctors about, epilepsy, what should we be doing? Nothing is working. And I started to educate myself. I started to learn that epilepsy is a spectrum disorder. You have some people who, like my daughter, are very, very severe. And you have other people that serve in government, have jobs, are celebrities. But it is a spectrum, and at that time, 20 years ago, we were only talking about the people who were doing well with epilepsy. We weren't talking about the severe end of the spectrum in the US. I also learned that it was really common. 1% of all of Americans suffer from epilepsy. So that's 3 million Americans, 40,000 in Ireland, 65 million people worldwide. In the US, 1 in 26 people will develop epilepsy in their lifetime. This is not some rare disease. There were so many, and why did we feel so alone? I learned that even though it was really common, and I had never met anybody, my daughter's story was not unique. 30 to 40% of all the people with epilepsy do not respond to any of the available treatments. I learned that more than 70% of people with epilepsy don't know the cause, and at a certain point, I think we stopped looking in the research world. And this upset me, this frustrated me. It made me feel like I wanted to help the effort. And I learned that the economic burden of this disease was huge. It's $22.6 billion per year in epilepsy. And that cost in the US is concentrated in the 30 to 40% of patients that don't respond to any of the available therapies. And so this made me want to get involved and get help. Something else that I learned was that the dollars that are contributed to epilepsy research are dismally low. This was taken from the US data where we calculated all of the public and private funding for epilepsy research in epilepsy, and you can see it's about $10 million per year that's spent. That equates to about $70 per patient with epilepsy in the US, whereas other diseases like autism's, Parkinson's, they're up above $50 million per year. And I think it's because we haven't told the whole story of the spectrum of epilepsy severity. There are patients that are not doing well, that are not living well, that really need some help. Something else that I learned is that despite the number of medications, more than 40 that have come out since 1850, we haven't made a dent, if you look at the research, in the number of patients that don't respond to these. So if you look back in 1881, 36% of patients didn't respond to the therapies that were available. Fast forward to 1971, where you have a number of new drugs that have come out, and this is just drugs. This is even diets or devices or surgery. Still, 33 to 38% are not responding to therapy. And then you fast forward to 2005, where the incidence, the number of people with epilepsy hasn't changed, and it's still at 38%. So we're creating all of these new therapies, but they're not making a dent in the population that would be affected by them. They're making a dent in the same people that would have responded to the previous therapies that were developed. So we have to change the way that we're tackling the problem if we're going to make a dent in our most severe, most expensive group of patients. Another thing that I learned was that we hadn't made a really big dent in whether or not you responded to a first treatment. So here's a study from Kwan and Brody in 2000 that showed that your chances of responding to the first seizure therapy is about 47%. 36% don't become seizure-free. But the chances of responding to the second seizure therapy is 13%. The third seizure therapy is about 3%, and then polytherapy is about 1%. So if the same group did the same study 12 years later and we really hadn't made a dent, again, about 50% of patients will become seizure-free on their first medication, about 13% with the second, and so on. So even though we have more than 40 different treatments that we can try for patients, the chances keep going down lower and lower and lower of responding to a new therapy that's added. And yet what we do with patients like my daughter and many of you who have children in this room is we just add therapy after therapy after therapy, hoping that something will work. And as a mother, the chance is never zero. of responding. The percentage is never zero, but it does go down. And so we need to do better. We need to get to the place where families don't have stories where they've tried and failed 20, 25, 30 different therapies, but that we get the right therapy to the right person at the right time, every time, which I'm going to talk about. And then I think the statistic that really just threw me over the edge was this one, was that between 1975 and 2014, we really hadn't made a dent in identifying the causes of epilepsy. And you're never going to cure it, you're never going to make a dent in it if you don't know the cause. And so this is the point where I started going to college. And I was determined that I probably wasn't going to help my own daughter, but we had to do better for the next generation of Savannahs out there. We had to start identifying the causes of epilepsy and getting those diagnoses to patients, and then we could begin to think about therapy and cures. And while I was going to school, so I was not a good student in high school, and it took me 12 long years in college. This was actually my PhD picture where Savannah is wearing my hat. And it took me 12 years, and I did fall in love with science along the way. I did not see my husband for 12 years. That's how we did it. He took care of her when I was at school, and I took care of her when he was at work. And he worked nights, and I went to school during the day, and we didn't see each other. But during that time, the thing that was the most compelling to me, the thing that could explain why a person could go from being perfectly healthy and typical and normal one day to the next day having horrible uncontrolled seizures that never gets back on track, was always genetics. And so I was drawn to genetics very early on as something that could make a difference in epilepsy. And for those of you that aren't very familiar or can't remember back to your early school days about genetics, every cell, nearly every cell in your body contains your DNA, which has about 20,000 genes within it. And those 20,000 genes make you unique and who you are, the way that they are formulated. And the way that I like to think about it is that you have a letter code for your DNA, and those letters can be in different order and different number. There's actually only 4 letters, but they can appear in a different sequence. And you have many of them. You have actually billions and billions of these letters in your body. And the best analogy that I've ever heard is it's like a library. So you are the library. I actually visited a really gorgeous library above the Book of Kells yesterday. It was gorgeous. So think about this library, right? You're the library, and your cells in your body are the books in the library, right? And the stacks and stacks of books. And the chromosomes, which the DNA is a part of, are the books themselves, and you have 46 of those. And the letter codes, the little letters, are the letters within the book that can be changed. And I think this is a good explanation. And so for each of us, that letter code within the book is different. Yes, we're all a library and we all are all books, but the letters within each of the books is different. And so as I was going on and getting my education and falling in love with science and becoming a geneticist, I started to think of my daughter more as somebody that I can learn from and study. And as a graduate student, I was working in a lab at the time that genetic technology had changed from being able to look at one gene at a time. We literally really looked at one letter at a time at one point, and then one gene at a time, and then a couple of genes. But the technology advanced to a point where you could look at all 20,000 genes at one time in a person, and it was becoming more and more affordable over time. And it's something that's still, even though it came out about 7 years ago, it's still very young and very new, and it's still very expensive and laborious, and it's still being done mostly in the research setting. But very slowly but surely, this is moving into the medical world. And this is something that we're doing here in Ireland with the groups, where they're trying to take it from a research protocol and learn who it will help and when to use it, and then one day in the future to move it into the medical space. For me, when this technology came about, I was working with my boss one day, and he said to me, how is your daughter? And I told him, I said, she's having 300 seizures a week. She's going into non-convulsive status 1 to 3 times a week. She's barely awake. She can barely talk. And I think that his response was really just something he said out of desperation. He said, let's enroll her in the study that we're doing on genetic epilepsies and sequence her exome. And I was like, okay. And so it was my job to sequence her exome. So I began at that point to start thinking about my daughter as a research subject and not just as my child. And up until this point, I had never really thought that I was going to make a difference in her life. As I said, I really went into this trying to help the next generation. So I drew this diagram. For those of you that have seen these family trees, the square at the top is my husband, the circle is me, the square at the bottom is my son, and the circle is my daughter, and she's shaded in because she's the one with epilepsy. And I began to do the genetic analysis. We did exome sequencing, which is what the name of the test is that looks at all of 20,000 genes. And begin to do the analysis slowly but surely, and I've sort of listed what that analysis would look like. And when I didn't find any of the known genes that cause epilepsy in her, I began to do what I call the crazy mother analysis of her genetic, where I started to group and cluster the genetic mutations that she had that were very severe and predicted to be damaging and very unique to her, and I didn't have them and my husband didn't have them. And what that led me to was actually something quite amazing. I'm all for the crazy mother analysis. I identified 10 different mutations in her that were affecting calcium channels. So here on your left, you'll see a calcium channel as it sits in the membrane of a cell, and you can see it's made up of a bunch of different pieces. And every one of those black dots there represents where she had a genetic change that was affecting the way calcium came into her cells, came into her neurons specifically. And all of those genetic changes were unique to her. Mom and dad didn't carry most of them. They weren't found out in thousands and thousands of healthy people. And they were predicted, in most cases, they were predicted to increase the amount of calcium coming into her cells. And in a few of the cases, we knew that those mutations, when made, caused too much calcium to go in the cells. So there's different levels of proof that these were driving her epilepsy. And then what was beautiful was that there was one piece of evidence in her life that only I knew. And that was that every time we put her on calcium supplements, when you give your child a vitamin, she would seize more. Always, every time. And all of our children go on these calcium supplements when they're on anti-seizure medicines because the anti-seizure meds leach calcium from their bone. So they always go on calcium. And every time she would seize more. And to me, that was the final piece, that piece of knowledge, knowing that putting her on excess calcium, giving her too much milk or cheese, really did upset the seizure balance. So there was something to this calcium factor. And so I began to start to make a case. And on the right side, I'll just show you that this is a neuron here on the right. And calcium is very important for the release of neurotransmitters on the presynaptic side down to the other side of the neuron. And the chemicals that are released when calcium goes into the cell affect the electrical signaling downstream in the brain. And so this is very important. If you have too much calcium going in your neuron, you have too much neurotransmitter being released, you have too much activity downstream, and it's an electrical storm. So I started to make a case for why we might want to try putting her on a drug that reduces the amount of calcium going into her cells. And it turns out that there are drugs already approved in the United States for patients that have heart defects, arrhythmias, and hypertension. And there are some actually very common, very safe drugs that are out there. And so I started to do what I call a risk-benefit profile. And I hope that 10 years from now, all of us mothers, fathers, doctors, nurses, and clinicians are using risk-benefit. We've never really had the scientific data to do risk-benefit, but you can start to generate statistics and start to look at whether or not going on a therapy is more beneficial, more risky, and then assessing as a family whether or not you think it's worth the risk. So I started to do a risk benefit, and my child was having 300 seizures a month. She was going into status all the time. She was asleep all the time. She could barely talk. We had no life. We never left the house. When we did, it was usually a catastrophe. She'd seize somewhere and pee all over herself, and we'd have to drag her out of whatever supermarket or store. And so to us, trying her on an FDA-approved drug that was used in millions of people in the U.S. had heart defects was worth it. We did check her heart, so her heart was healthy and functioning properly, and we've checked her heart ever since putting her on this medication. And we put her on it 5 years ago, and this is what the graph looks like. You could see that she was having between 100 and 300 seizures a month in the 2 years prior to going on this drug. It's called verapamil. It's a blocker of L-type calcium channels. And we had immediately, within a week, a 95% reduction in the number of seizures. and no other therapy had ever had that impact. This was the diastat use before and after verapamil. Widow. Thank you. Sorry, it was hell, as you guys know. So the diastat use, the non-convulsive status, please. 5 years. 5 years. So while we haven't completely stopped her seizures, she still does have seizures. She's been overall a 98% improvement in her number of seizures, and more than that, 99.9 for the verapamil, for 5 years. And so any of you that have ever experienced a honeymoon on an anti-seizure drug, you know that 5 years is pretty amazing. And in fact, we didn't talk about it for a year. I was like, she's doing really well. We're not allowed to talk about it because I'm not sure if this is a honeymoon or what this is. I'm convinced, though, now, because I'm also a researcher and I've seen a lot of data come out since then, that if we had put her on this medication earlier, that we actually probably would have changed her life and reduced her seizures. And it's not because verapamil is a good seizure drug. My daughter has Lennox-Gastaut syndrome. I don't think Lennox-Gastaut patients should go on verapamil. I think Savannah needed to go on verapamil because of her unique genetic code that made her who she was. And that's the point of precision medicine. You know, I think if I had heard this story 15 years ago, I would have been out looking for verapamil for my child. But the reality of the situation is it could make somebody worse who has too little calcium going into their cells. And so you have to really understand that. And we have to get to this place where we're looking at the genetics of people and we're truly understanding the biology the best that we can as we move towards precision therapy. So in a way, we were lucky. It was a calculated risk, but in a way, we were very lucky. And this is a picture of Savannah today. Let's see if I can get it to load here. There she is. So you can see. So you can see she's much more awake and engaged. It's not perfect, but I really do feel like we're meeting her for the first time in our lives. She's sassy and silly and oh my God, is she bossy? She is so bossy, and she's very funny. She has a wonderful sense of humor. And she's really having to learn things that she never had to learn before. Like you can't, throw a tantrum in order to get what you want. You now have to, behave responsibly. But she can get her words out. Epilepsy would always trap her words in her head or the medicines. She's come off, she was on 7 medications at the time that we started verapamil, and now she's on 3, and the diastat is gone, and we're slowly coming down on those. And so I really think that the most surprising thing for me as her mother is while I had never given up hope, I was cautiously hopeful, right? What if she was going to have seizures for the rest of her life? I just never thought a therapy in an 18-year-old could change your life as dramatically as this changed our lives. In fact, the fact that I'm in Ireland at all and not home caring for my sick child is pretty huge. And I've never been happier to be wrong, actually. Never been happier to be wrong. And so we can actually intervene in adults too. It won't be perfect, but I think Savannah's story not only says we should be looking to genetics to guide therapies in our research, but it also says that it's never too late to intervene with the right medicine. And if we can just get enough research to move it forward, then I think we can achieve this in the future. It's not going to be fast enough, and we all have to play a part in it, but I do think it's going to change every aspect of the way that we diagnose and treat patients with epilepsy, and not just epilepsy, all diseases, because your genetics defines everything, every cell in your body, from the way that you metabolize food and whether or not you get rashes on your skin, tumors, ulcers, things like this. So I think all disciplines of medicine are going to be looking to genomics in the future. So let me just talk for a few minutes about the genetics of epilepsy and when I became interested. So initially, and this is some people in this room who've done some amazing work on this, Initially, the first genes for epilepsy that caused epilepsy that ran in families were identified in the mid-90s, and we identified a few genes in the first few years, and we thought that that was going to be a revolution at that time. This is back when we were looking at one gene at a time. And then we had a period of time which we call the Valley of Death, where despite many, many studies, we really didn't find, as researchers, very many genes in epilepsy until the technology changed. And the first technology to come on the scene was copy number variant changes, where you could start to look at the number of copies of a gene, right? So you're supposed to have 2 copies, but what if you have 1 or 3? And you might think of this as Down syndrome, where the patients with Down syndrome have a completely extra chromosome, right? So copy number technology came along first, and then next generation sequencing came along, and this is the technology I was talking about, where you could sequence many, many genes at one time. And some groups, I know here in Ireland, they're sequencing a panel of genes, not all 20,000, but a group of them that are all known to be associated with epilepsy. And then, of course, you can sequence all 20,000 genes. And then as soon as it becomes cost effective, we'll be able to sequence genomes, where you sequence all of the letters, the billions of letters in the letter code, and not just the ones that code for the genes. And that's going to be a little while in coming as we try to get the cost down. But it's exciting to think about that, how this is going to change the future 10, 20 years from now for everybody. So how many genes are there in epilepsy today? Well, epilepsy, it's been an explosion, and Sam Berkovic, a lead researcher in Australia, says that there are more than 300 genes now that are associated with human epilepsy. There's a little over 20 that cause inherited single gene epilepsies. There's more than 300 that cause the structural and metabolic epilepsies. And we're pretty inclusive. Any disease that has seizures, we group into the epilepsies. And so these would be your chromosomal syndrome. Some Down syndrome kids, for example, have seizures. These would be your cortical malformation kids, your mitochondrial diseases. And I think what researchers found was surprising was that we would find these things we call de novo mutations. So these are mutations that mom and dad don't carry, but the child does carry, and they're causing the person's epilepsy. So this is inherited. It's not inherited. It's after the sperm and egg come together, and you have 1 cell, and then 2, and then 4, and then 8. A mutation develops at that point. And so the mutation is genetic but not inherited. And so for the first time, those two terms have been separated from each other. And my daughter, many of her mutations are actually de novo. And what we found more than 26 genes that cause, this is actually an old slide, there are more now, that cause the very severe pediatric onset epilepsies like my daughter Savannah has. And it's proving now that, I'm going to show you some data in a minute, that we can solve about 30% of these very severe pediatric cases by looking for de novo mutations in these genes that we know cause epilepsy. So here's a population that that we haven't made a dent in, that is extremely expensive, and we think that we know the cause in 30% of them. And the research is coming out steadily that every population that's studied, whether it's in Ireland or in the US or in the UK, that we're solving 30% of our very, very severe kids. Now, this is still a very small percentage of epilepsy, but it's a percentage that we've not made a dent in. These are your Lennox-Gastaut kids and your Dravet syndrome kids and your very, very severe epileptic encephalopathy kids that have their futures completely derailed by epilepsy if they live. There's a lot of variability. You can have genes that cause the same gene cause a very severe epilepsy and cause a very mild epilepsy, and we're just beginning to scratch the surface on this. And this is why I say it's not going to be fast enough for us, because even though we can sequence every one of you in this room, we could sequence you today, but it doesn't mean we would know how to interpret it, even if we had the money to do it. And so there's a lot of work still to be done. But Dr. Berkovic, who's one of the lead genetic researchers, believes that most of the epilepsies in the 70% that are unexplained are going to have a strong genetic component to them. And even the epilepsies that are acquired by some sort of a trauma probably have a genetic component in place. And so the genetic revolution is really changing everything and the way we think about it. And so it introduces this idea of precision medicine, previously called, individualized medicine. or genomic medicine is the new one. And we're way over here on the left where we're still doing the profiling of everybody's genes, but our goal is way over there on the right where we separate people out by their clinical picture, by their other tests that they have, for example, spinal taps and what's going on in their spinal fluid, and their genetics, and then stratify them so we know which drug. And that is our goal, is to get to that place. So what about clinical genetic testing? The experts in the field actually already have a recommendation for our kids. We just haven't done a good job of explaining it to the world, even in the US, even in the UK, no matter where you are. If a doctor such as Norm here is seeing a patient and he suspects that there is a specific epilepsy, he should test for that gene. If it looks like Dravet syndrome, experts in the field of genetic clinical medicine say he should test for that gene. He can't get the test, insurance won't cover it, it's too expensive, but the experts say that this is where we're going. If it's a nonspecific epilepsy, you don't know what gene it is, then maybe you should do a gene panel, which are very important. This is where you take a handful of genes. I've seen panels that are 100, 200, 1,000 genes that all have some implication in epilepsy, and you look at those. And this is much cheaper than doing all 20,000 genes at one time. And also copy number testing. Remember the number of genes that you have. If you have one too many or one too few, this is a problem and can cause epilepsy. And then there's whole exome sequencing, as we talked about, and this is where you look at all 20,000 genes at one time. And the beautiful thing about whole exome sequencing is it's not necessarily better than any of the others, but if you have the sequence for all 20,000 genes, you can now also do additional research on the genetics in those people. For example, what if there are two genes that are causing a person's epilepsy, the double hit hypothesis, they call it. You can do that type of research. Or what if there are genes that regulate your response to medicine? We actually know there are, so they call them pharmacoresponse genes. And so you can start to do research on that. And we've actually begun an initiative at Cure called EGI, where we're looking specifically at exomes, which I'm going to talk about in a minute. But before I do that, I just want to give you just a tiny bit of data. I think many of you have heard about the Epi4K consortium, or some of you have. This is where they sequenced 4,000 epilepsy exomes and genomes in a group, and I know that Norm and JP are a part of this group. But in this group of 356 parents and their child, a trio, with severe pediatric onset epilepsy, were able to solve 17% of them. So find a gene and 70% right up front. And then there's another 25 to 30% that they think we're going to get solved. And what we found, there's another initiative going on in Europe, Euroepinomics, is that when they published their large data set of patients, that many of the genes that were on the watch list moved to these are epilepsy-causing genes. So we think the yield is going to be 30%. That's just one study. This was another study done by the largest genetic testing company in the US, GeneDx, and they presented this at the American College of Medical Genetics meeting in 2015, where they did 40,000 pediatric epilepsy patients. When they did panels, gene panels, where they just looked at a group of genes, they were able to find the cause in 22%. And when they did exomes of mom, dad, and child, they were able to solve 40%. And these are just two of many studies that have come out where they are showing that somewhere between 30 and 40% of patients with these severe pediatric onset epilepsies are solvable. So this is the gene that is really driving your epilepsy, not you have this gene confers a 1% risk factor. And so we're still figuring out who to do that in and when to do it and how we're going to pay for it, but I think it really it speaks to how we're going to change the way that we diagnose. And that's identifying the cause, and we often talk about, if there's a cause, is there an associated therapy, a treatment associated with it? And there are some, there are very, very few, and they're all experimental, but there are a number of other things that happen if you get a cause. For 16 years, 12 years of college, 16 years of agony, I searched for the cause of my child's epilepsy. I never thought it would make a difference, but I did. I was always looking. And until you get that as a parent, you're sort of stuck. And so I think just knowing the cause is important, but there's so many things that can happen if you have a genetic diagnosis as well. You can get connected with other families. You can begin to look at your kid in relationship to somebody else's child. We have one of my favorite stories is a group in the US that all got together with a newly diagnosed epilepsy, SCN8A epilepsy. And the parents did a survey amongst each other, and I think it was only like 19 kids that had this disease, but almost all of their kids, I think all but one, got worse on one specific epilepsy drug, which they then told researchers who then researched it, right? And it's the opposite drug that you would expect. So the drug was levetiracetam, which is a drug that you would typically put another group of kids with an SCN mutation on. And so it was counterintuitive, and it's really guiding researchers. And so it's exciting to think about parents getting together and actually generating meaningful information that researchers might be able to use in their labs. It ends the quest for the diagnosis. It allows for reproductive planning in many cases. If you know that you have a de novo mutation and that you're not carrying that mutation or your child has a de novo mutation and you're not carrying it in any % of your cells, then the chances of you having another kid with LGS is very, very low. It's not zero, but it's very, very low. The other thing for us was I have a son, and I've always been fearful that he's going to have a kid with LGS. And so if you get a genetic diagnosis, we can begin to start talking about genetic counseling and whether or not you stand a risk of passing this on to other children, or your children stand a risk of passing it on. And it ameliorates the guilt or shame associated with it. I think if you have a genetic diagnosis, it's not your fault. You didn't sin. You didn't upset Buddha. I mean, what all of these things that we hear as parents that reasons why our kid had epilepsy, whatever they may be, it's a biological abnormality. And I think that if we can diagnose this as such, there are many parts of the world that still believe that epilepsy is spiritually caused, witchcraft or demon possession. And so, moving it to a molecular disease is important. And then connecting to community cannot be underestimated. We just counted recently that there are 31 gene-specific epilepsy groups that have formed around there. I told you there are more than 300 genes, but there are 31 groups that have formed. They've started their own gene-specific group. For example, the SCN8A Epilepsy Foundation, the Dravet Syndrome Foundation, the Lennox-Gastaut Syndrome Foundation, and they're all forming, and they're finding each other, and they're raising money for research, and they're talking to each other about their kids and about quality of life issues, and I think it's beautiful. I think it's beautiful. So if we're going to get to this precision medicine place over here on the right, we need to tackle this place on the left. And I'm going to just spend my last 2 minutes talking about the Epilepsy Genetics Initiative that we've started at Cure, where I work. And this is really an extension in my mind of trying to do what I sort of accidentally did for my daughter, but more broadly to the rest of the world and a much more scientific way. So EGI is a database of sequences that will be exome sequences from patients with epilepsy that is going to be stewarded by Cure, a non-governmental organization that's run by patients and parents and advocates. We're going to take that exome data. Most of it's generated in the genetic testing labs, and research isn't performed on it. We're going to analyze it every 6 months, and anything we find we'll report back to the treating physician. And by anything we find, I mean any known causes of epilepsy. And the reason is because you have to analyze this data every 6 months. If you had exome sequencing done 2 years ago, you haven't been looked at for all the new genes that have been discovered in the last 2 years. And so we have to regularly reanalyze this data because the state of genetics research is accelerating and is going so quickly. So we're going to report what we find, known causes of epilepsy, back to the patients. And then we're going to de-identify the data, take off everybody's name and identifying information, and give it to researchers so they can start looking at all those other things I talked about, genes that might regulate pharmacoresponse or genes that might be contributing to the epilepsy. Why is it the 2 kids with Dravet syndrome that have the exact same mutation don't look the same clinically? They have different features. Is that genetic? And so all of this research that we can be doing. So I'm going to take a minute just to show you a really short video about EGI.
Imagine having the ability to unlock the genetic secrets of epilepsy. Just think of what this would mean for the 65 million people currently living with this debilitating disease and the doctors who struggle to find a way to stop their seizures. Knowing what causes epilepsy and what treatment might work best seems almost like an unattainable dream. But thanks to modern advances in genetic research, this dream is close to becoming a reality. By examining the changes in a person's genes, researchers have been able to identify altered genes that lead to some people's epilepsy. With about 20,000 genes in each of us, this is no easy task. However, by using genetics to diagnose and treat each person individually, we could create a world where we tailor treatments to each individual person with epilepsy. Unfortunately, current gene testing is limited because studying gene data is challenging, and analysis sometimes needs to be repeated again and again over time before we can identify a cause. Now, that's where we come in. The Epilepsy Genetics Initiative, or EGI, is a project of CURE dedicated to advancing epilepsy genetics. CURE has created a place where people with epilepsy, with the help of their doctors, can house their genetic data to have it analyzed and reanalyzed to help find the cause of their own epilepsy and help advance science as it seeks to uncover new causes of epilepsy that have yet to be discovered. Researchers will be able to study the genes which will be confidentially collected in EGI and build a database that has never existed before. Just imagine what this will mean for epilepsy research, therapy development, determining which treatments work best for which people, and discovering why some people with epilepsy suffer from coexisting conditions like depression and learning and memory challenges. By bringing together people with epilepsy, medical professionals, and scientists, EGI will help to improve the diagnosis and treatment of epilepsy in a way that has never been done before. Find out more at www.cureepilepsy.org/EGI.
So this is an initiative that we've started at CURE, which is really exciting, and I hope you'll take some time to go look at the website. And in addition to analyzing and trying to help patients to get to their genetic diagnosis, and right now it's just a handful of patients that have a genetic diagnosis compared to all the number of people with epilepsy, we're also trying to educate the community broadly about the different genes that are out there. So please visit the website. And I just want to say that even though it will be slow, and even though there won't be a genetic diagnosis for most of us in the room, and even though if there is a genetic diagnosis, there's probably not a treatment that's available today, that's the future of where we're going. And when I look back 20 years ago on epilepsy and my journey in the last nearly 21 years, I've never been hopeful until about 5 years ago when I started to see the way genetics has changed the way that we do medicine. So I really do think that this is going to come in and it's really going to change things. And as somebody who has a 22-year-old who still hears stories that sound exactly like the journey that we went through, of the severe diagnosis and treatment journey, I do think that the day will come to an end. But it's going to require all of us working together, doing what we can, doing our part, and helping to push this effort along. And I know it's already going on here in Ireland. So I know Norm's probably freaking out. I went over by 5 minutes, but I'll stop there. Thank you.